Good afternoon. I'll be talking about potential biomarkers for treatment response in PCV. Basically, PCV has a variable presentation starting from chronic CSR to massive subretinal hemorrhage. The treatment responses are variable, often disappointing. And determinants to treatment could be either genetic profile, choroidal morphology, or possible other biomarkers. The genetic associations have been associated with the ARMS2 HTRA1 locus as well as the CFH locus. They tend towards either similarities to ARMD or CSCR. And our understanding of the Pachychoroid spectrum tells us that CSCR and PCV could be two ends of the same spectrum when the choroid is thickened. The subtypes based on morphology could be either the RMD end of the spectrum where the choroid is thin, response to anti-VEGF is poorer, is better, and there is an ARMS2 HTRA1 association, whereas typical PCV has a thicker choroid, better response to PDT, and has an association with the CFS gene. So we looked at other biomarkers, more importantly regulatory molecules that could drive our treatment response. MicroRNAs are basically a class of non-coding RNAs important in controlling gene expression via regulation of protein coding genes. They occupy 1 to 5% of the human genome and regulate at least 30% of protein coding genes by either degradation or translation inhibition. And they have been shown as a biomarker and regulatory of disease-causing genes, both in DME and AMD, and as of yesterday, there has been no report on PCV. Similarly, for long non-coding RNAs, they are more than 200 nucleotides long, have no apparent protein coding potential, but they can regulate various biological proteins by chromatin remodeling or protein translation. And they have been recently showed as newer regulators of angiogenesis in retinal neovascularization, including diabetes and ROP. So our research questions was, is it possible to identify PCV disease specific and drug response specific microRNAs and long non-coding RNAs in cases? And could these be used as biomarkers for screening or potential therapeutic targets? This was a pilot study for feasibility. We took 4 cases. One was treatment naive, one was active and unresponsive after 6 monthly injections, one was on PRN maintenance treatment, one was a normal control. For equality, all were females with no major systemic comorbidities. And this was the technique. We isolated RNA from the blood sample, sequenced it to get the small RNA library, and then compared it with the referenced human genome and assessed the known and novel mRNA prediction. And then also we looked for the microRNA and long non-coding RNA sequences and validated with qPCR. And these are housekeeping microRNAs that are formed everywhere in the human genome. What was surprisingly that the biomarker for colorectal cancer was seen in all our cases. The significance is not known. The top right shows you housekeeping microRNA from the LET7 gene, which is seen in all humans. Now coming to known microRNA expression, the treatment naive cases showed a very high tag count of microRNAs known to code for cell migration and proliferation, and as well as a very high tag count of known microRNAs known to be responsive for tumorigenesis. This is the heat map showing the relative tag counts of the various microRNAs. And we found unique microRNA expression in all categories. It was the highest in the treatment naive category. Similarly, we found different long non-coding RNAs from all categories, and this was the overlap Venn diagram. So to conclude, it's the first report proposing the role of microRNA and possibly long non-coding RNA as biomarkers to determine treatment response in PCV. We found differently expressing microRNA and long non-coding RNA in each category, and discovery of novel microRNA is not described so far. Since submitting this paper, we have expanded it. We have now 3 cases in each category. We have found similar differences in expression, and validation is underway. Thank you.